chief resident of radiology over in Congress. There will be images on this test. What you've had so far, what she's going to present, there'll be some images on the test. I know that. The second thing is, I don't know, I wasn't aware of this, but you know, when you go to a Caribbean island and the beaches are pristine, you know, the picture, that's only because somebody was there during the night to rake up the plastic bottles and the rubber tennis shoes and the trash that's in the ocean. Every night that has to be cleaned. The beaches have to be swept. So I would appeal to everyone to never use a plastic bottle again. It's just amazing how much in beaches that don't get swept. It's amazing how much trash there is in this ocean. Or recycle, definitely. You know, sometimes, well, I was looking forward to last night not doing anything because it's been a long time since I've not been able to do anything at night. And I had a visitor about 8 p.m. that stayed until 11. It was just, you know how sometimes you get a visitor and it's just bizarre that they show up. This was one of those instances. It was a good visit, but it's like, wow. Never mind. Okay, so anyway, we're going to talk, Kimberly Wood here, she's a guest today. So we're going to talk today about the cerebellum and the cerebral cortex, and then a little bit more about the limbic system. We're going to beat that horse to death today. So, these are the lecture objectives. We're going to start with the cerebellum, and we'll work our way up. When you're talking about the cerebellum and its interplay with the rest of the nervous system and movement, you see here, this is a rather simplified, but it's a rather accurate diagram of movement. And obviously, if we're talking about cerebellum, we are talking about movement. If you go to the cerebral cortex, the pathway that you're quite familiar with is the corticospinal tract here. But you know that the cerebral cortex provides a great deal of information. If you go back to the basal ganglia lecture, that's one of the afferents into the basal ganglia, which is going to project then to the thalamus, which projects back to the cerebral cortex to modulate activation. You also get other areas of the brainstem involved. Remember, we didn't talk about it, but the inferior olive in the medulla, we have the pyramids, and there's the pre-olivary sulcus, where cranial nerve 12 is, and then there's the post-olivary sulcus, where the olive is between them. It's a large nucleus. If you look at a cross-section of the medulla, it's that, this is the, so here are the pyramids here. It's that squiggly line that you've seen on the cross-section. That's the inferior olive there. The inferior olive has a huge input from the cortex into the cerebellum. That's one of the main pathways where you get cerebral input regarding motor function into the cerebellum. The cerebellum, what it does, it's seen right here. The overall bottom line purpose of the cerebellum is to perfect timing of movement. And it does so because the proprioceptors that you have in your limbs, in terms of where your parts are in space, a lot of the dorsal column pathways regarding proprioception make their way directly to the cerebellum. So you can't time movement unless you know where body parts are in space. And then, of course, the basal ganglia and the cortex. And there you go. Okay, one of the things here about the cortex that you should, it says control of fractionated movements. In other words, fine movements. Fine movements. That's what that means. The functions of the cerebellum, overall three: equilibrium, with its association with the vestibular nucleus, and it's linked into vision, and that's why you have a Romberg reflex, right? A Romberg test. Posture is mostly related to the exoskeleton, and then modulation of the appendicular skeleton for timing of movement of the limb. And intended movements, it says here, movement is not just pure, okay, here I go. You have to generate intent, and then you have to develop a plan for movement, and then you actually have to execute it in the proper timing so that I don't overshoot that finger to nose and so on. Sophie, what in the hell are you eating?
Peanut butter, toast, and a banana.
That is most amazing. That girl weighs 40 pounds, and she, every time I look up, she's eating something. Okay. If we look at the midline section through the cerebellum here, you see two things. You see the white matter right here. The white matter is called the arborvitae, like a tree, look like a tree. And then you see the gray matter around it. And this is the perfect section right through the middle of the cerebellum. There are two lobes of the cerebellum, a right and left lobe. And in the midline, you have what's called the vermis. The vermis. Interesting, this is from the bottom of the cerebellum. So what is that hole right there? That's the brainstem. What is that hole at the bottom? The foramen of what? Magendie. It's the one in the middle. That's the foramen of Magendie right there. Okay? If you look at a transverse section through the cerebellum, though, you'll see that you get much more than just the arborvitae. You have the two lobes, and here's the vermis. You actually have the hemispheres, and then deep within and buried within the white matter of the cerebellum, you have 4 cerebellar nuclei. Just like in the cortex, you have the deep brain nuclei, the thalamus, hypothalamus, basic ganglion. You have the same thing in the cerebellum. You have a place where input goes, and then output leaves from there. So you don't have a lot of direct output from the cerebellar hemispheres to the outside. It has to go through one of these deep cerebellar nuclei, just like most of the cortex goes through the basal ganglia to affect movement. It's attached to the brainstem by 3 peduncles, the superior, middle, and inferior peduncle. Different tracts go through different peduncles. Remember the dorsal and ventral spinocerebellar tract? They go through one or the other. I don't want to get into all the tracts. There are about 20 tracts that go in and out of the cerebellum. But the main input to the cerebellum, as you already know, comes through the pons, the pontine nuclei, through the middle cerebellar peduncle. The middle cerebellar peduncle is by far the largest of the 3 connections, and it contains solely corticopontocerebellar fibers. Corticopontocerebellar fibers. That's the cortical input into the cerebellum. Okay? Through the pons. Any questions about that? Now, here's the, if we dissected away part of the cortex, you can see these nuclear groups. They're pretty well defined in a dissection. There they are. There are 4 of them, as I mentioned. They're called the vestigial, you don't need to memorize these, the vestigial, globose, emboliform, and the dentate. The dentate being the largest. The dentate looks a lot like the inferior olive of the brainstem. So when I ask you what is the output of the cerebellum, you would say the deep cerebellar nuclei. I remember, let's see, okay. So if we look at this illustration here, the cerebral cortex and the cerebellum are constructed the same way. The white matter is on the inside, the gray matter is on the outside. That's opposite of what you see in the spinal cord. So you can see the white matter here, the gray matter is on the outside. Whereas in the brain, you've got gyri, in the cerebellum, you have folia. The folia are the gyri equivalent in the cerebellum. You can see here a white matter tract. You can see the layering of the cerebellum right there. Now, I don't want to get into a lot of the circuitry of the cerebellum because there are a bunch of cell types in the cerebellum and in the cortex. So you're not going to need to know the circuitry specifically. The points I want you to know about the circuitry I'm going to emphasize. Okay? One of the points is that the functional unit of the cerebellum is the large Purkinje cell. This is one of the largest cells in the nervous system. And the Purkinje cells, you'll see, they line up in part of the gray matter of the cerebellum, along the folia. They're lined up. You can see them lined up right here. Now up there in that illustration, here's the white matter, here's the gray matter. The gray matter is actually layered. It's not just uniform, it's layered. So you can see different nuclear clustering along the gray matter. If you, so if you look at what cell types are there, the Purkinje cells occupy one layer, and then there are all sorts of interneurons connecting the Purkinje cells up here and down here. The large Purkinje cells have one axon, but their dendritic tree is freaking massive. It's huge. There's one axon coming out of that little thing right there. That axon is headed for the deep cerebellar nucleus. And it is inhibitory. Now the thing about this huge dendritic tree here is that they're all oriented perpendicular to the plane of the folia. So if the folia is running that way, you have all of these Purkinje cells lined up like that. They're all lined up the same. As you see here, this is viewed 90 degrees from each other. There you go. Okay? The input to the Purkinje cells from the rest of the nervous system comes in two forms. There are climbing fibers and there are mossy fibers. And this illustration shows you all the different interneurons that you don't need to worry about. We're going to focus on the climbing fibers and the mossy fibers. If you look at if you consider the climbing fibers, what these guys do is that, first of all, they come from the inferior olive. That's the only place they come from. So different motor systems project to the inferior olive. The inferior olive projects to the Purkinje cells as climbing fibers. The reason they're called climbing fibers is that you see up here on this illustration, you have one fiber coming in, and it's going to essentially thread its way up through that massive dendritic tree, synapsing on that Purkinje cell 1,000 times or more. So you have one climbing fiber making 1,000 synaptic contacts on one Purkinje neuron. Now, remember when we were talking about activation or EPSPs and the development of the action potential You know, just one stimulation usually doesn't generate an action potential. You have to have a whole bunch of inputs in order to generate an action potential. Well, if you look at climbing fibers, you've got one cell hitting this neuron in terms of an action, a synapse, 1,000 times. The Purkinje cell is going to fire with that much input. But only one Purkinje cell fires. So what I'm telling you is the climbing fiber system is very specific input in and very specific output out. Versus the Mossy fiber system, where the Mossy fibers come from everywhere else in the nervous system. Every other input from the nervous system into the cerebellum is a Mossy fiber. And what these dudes do is they come in there and they synapse on one Purkinje neuron, and then go synapse on another Purkinje neuron, and another one, and another one. They have one synapse on 1,000 Purkinje neurons. So the one Mossy fiber firing is only going to synapse one time on a Purkinje. That's not going to activate that monster Purkinje neuron. In order for Mossy fibers to activate a Purkinje neuron, you have to have thousands and hundreds of thousands hitting it at the same time. In order to do that, you have to have massive input from a variety of sources. It's very diffuse input. And the output, because it hits on hundreds of Purkinje neurons, is not going to be fine, but a huge output. That's the difference between climbing fibers and Mossy fibers in terms of how they impact the cerebellum. You get the drift? Okay. All right. If you look at these things here, here's a Mossy fiber, there's a climbing fiber. The output from the Purkinje neurons is GABA. It's inhibitory to the deep cerebellar nucleus. Oh crap, wait a minute. How does the cerebellum then modulate movement if these guys, the functional unit of the cerebellum, is to inhibit the nucleus? Well, it goes back to the thing of disinhibition. The nuclei, deep cerebellar nuclei, they fire like crazy. What you have with the Purkinje cells is slowing them down. They're putting a brake on the outflow of the cerebellum. If the cerebellum were allowed to just go rampant, what happens to movement? You can't control it. And because this is associated with timing of movement, the more you try to time movement, the worse the tremor gets. That's how they work. See? It's easy. Because these Purkinje cells are lined up in a row, it's like the cortical columns that we discussed with the visual cortex. The same thing happens in the cerebellum. Certain parts of the cerebellum control certain parts of movement, and it's very specific to that movement, whether it's flexion, extension, rotation, pronation, supination, anti-gravity, fall down on your face, whatever. It's very specific. Take-home messages here is cerebellar inputs have place-specific information, like proprioception. That's place. I know where my feet are. I'm not having to look at them. But what it does is it takes place and converts it into timing so that I can walk without having to look at my feet. Any questions on the cerebellar dysfunction? Cerebellar intensive tremors we've gone over. And so testing for cerebellar function is, as I've told you I think it's a long time ago, but actually it was just last week. Get the body in movement, that's all you gotta do. Pass pointing, heel down to shin, get the body moving where timing errors can manifest themselves. This diadochokinesia is just your inability to perform these movements. You're gonna see that word on the board, I can promise you that. You might as well remember what it means. It just means that you cannot sit there, if you have a cerebellar lesion, you cannot sit there and do this. If you drink a 6-pack tonight in succession, you have dysdiadochokinesia. Okay? Nystagmus we already talked about in reference to the vestibular nucleus and its role in Romberg and other things. Okay? Got to remember though, that the single largest input to the cerebellum is through the pontine nuclei. Single largest input to the pontine nuclei is the cerebral cortex, which we're going to talk about now. Okay, so the cortex, cerebral cortex, is the gray matter on the outside, and you can see it layering of cells. And between these layering of cells, you're going to have axons and dendrites of other types of interneurons. And there are a lot of interneurons in the cerebral cortex. The white matter tract out here is eventually going to make its way to the internal capsule, right? It's eventually going to make its way to the internal capsule. The blood supply of the cortex, we know, you've had this now several times, anterior, posterior, and middle cerebral arteries coming from either the posterior circulation. So if you have a posterior circulation stroke, vertebral, basilar, posterior cerebral, you're going to have problems with balance because it affects the brain stem. It supplies the brain stem and vision. If you have a problem with the middle cerebral because it supplies the dorsolateral surface of the brain, you're going to have a problem with motor and sensory. Anterior of the knees up. Anterior cerebral artery supplying the midline, you're going to have problems with motor and sensory related to the knees down, from the knees down. So you have, you know, the Brodmann's numbers here. The Brodmann's numbers, 1 through 44, he designed these numbers based on the architecture. One of the general aspects of the architecture is if you look at the gray matter, it comes in 3 varieties. It comes in a 3-layered piece, and that's called the archicortex. The paliocortex is 3 to 5 layers, and the neocortex is 6 layers. The neocortex is what humans have the most of. It says 90% of your cortex is neocortex. You can look at these things as 3 different levels of evolutionary development. Neocortex is higher cortical function, regardless of what it is. Either calculation or vision, that's higher cortical function. The paleocortex is old, like the transverse gyrus epithelial. The is that model. The archicortex is the limbic system. That's the oldest phylogenetically part of your brain. And that, as you know, is the emotional side, or the emotional aspect of the CNS. Okay. These stains stain different types of things. This one right here is will stain cell bodies and a lot of the processes. Of course, the functional cell type of the cerebral cortex is the pyramidal neuron. The Nissl stain stains basophilic substances within the cell, and those basophilic substances are not out in the processes, so you really only get most of just the cell body in the proximal processes with the Nissl stain. And then Weigert stain does nothing but axons. Oh, but one thing I was saying, something I forgot to tell you last week. Talking about myelin and wrapping, going back to that lecture years ago. And that when you are born, your axons are not all myelinated. They actually don't, it takes a couple of years, actually several years, before you myelinate the axons to your adult condition. That's why little kids have to pee in their pants. And they will continue to pee in their pants until they myelinate their axons down to their parasympathetic nucleus at S2-4, which takes 2 years. That's why it is impossible to potty train a kid at 1 year or a year and a half, because they're not, they cannot neuroanatomically control their bladder. And that's one of the reasons why people think that your brain is not emotionally matured until you're about mid to late 20s. That's why people in college act so stupid most of the time, because they haven't finished maturing their nerves. It won't be on the test, but it's the point I wanted to make. That's why if you ever get asked by an attending, why is it that you can't potty train a kid before, 23 months? Because neuroanatomically, they can't control the sphincters or their trigger muscle. I just forgot about that. Pyramidal cells, they look like a pyramid to somebody. They look like little triangles to me, but I guess three-dimensionally they look like a pyramid, so somebody says they're called pyramidal cells. There are tons of different types of cortical cells. The pyramidal cells, remember, have just one axon, but unlike what you see in the cerebellar cortex, and pyramidal cells have a lot of collaterals. They have a lot of collaterals. So if we're talking about the corticospinal tract, those pyramidal cells in area 4, right, of Brodmann come out, they form the cerebral peduncles. And what most of them do is they send a collateral into the pontine nuclei, and then the main axon continues down as the pyramidal tract. So there's a lot of collateral input into those pontine nuclei. They don't just stop there, they relay through there. Brodmann's areas 1 through 44 or so. So here are the ones that are kind of significant that you should know what they are. We know about area 312. You see, I told you they still do it 312. I don't know why they do it 312, but they do. As the primary somatosensory area, that's what synapse is there?
Tertiary?
Where is the input to area 312 coming from? Coming from thalamus, right? It's coming from thalamus. If I do that, what nucleus of the thalamus relayed that information?
BPL.
UPL. If I did that, what information? It kind of actually hurt because it's on my earlobe. What information relayed the information to 312?
BPM.
Okay, good. All right. Area 4 is the primary motor area, precentral gyrus, right, versus postcentral gyrus as 312. Area 6 is called the premotor cortex. Area 6 is right in front of 4. There's area 6 right there, because 4 doesn't intend movement. 4 has to be told to move. Right? The corticospinal tract is originating from 4, largely. But it just doesn't say, okay, I'm going to pick up Andrew's mouse. So I want to pick up Andrew's mouse. That desire originated somewhere in my cortex. The initiation of movement did not start with area 4. It started with area 6. Area 6 said, you're going to pick up his mouse. These are the areas of area 4 that need to come into play to do that. It's the right upper extremity. Right? So area 6 actually drives area 4. Area 8 is called the frontal eye field. Dr. Morgan and Wheeler yesterday, they mentioned this area here because the frontal eye field is important for coordinating your eye movements. Saccadic movement, like being able to move your eyes back and forth. You have to be able to do that perfectly together. If they don't move perfectly together, then what you wind up with is double vision, diplopia, right? So the saccades, or saccades, however you want to pronounce it, this is the frontal eye fields. That's what controls that. So they were talking about nystagmus, and your eyes rolling to one side. You know, if you have a lesion on the left, your eyes will roll to the left. Point to the right, but you know what I mean. Your eyes will roll to the eye, or point to the side of the lesion. That's what the frontal eye fields do. Area 5 and 7 is a somatosensory association area. What's the difference between the primary areas and the association areas? The primary area is where that information is received. The association areas are adjacent to it and are areas where the sensation is interpreted. So when I did this, that was area 312. Where in area 312 was it? You got to go back to the homunculus. Where is the forearm on area 312? It's right out here, right? Correct? Well, that information went somewhere beyond 312 because I can tell you all sorts of information about that. One is it felt like a stick and not a metal rod. 2, it was room temperature, not hot, not cold. 3, it was a crisp wrap, but not painful. I could give you 100 descriptors of that. Those descriptors have to be in the association areas. And the association areas are fed with your memories. The association areas are fed with your memories because otherwise I couldn't tell that was wood. Right. Primary visual area we talked about. This visual association areas are the adjacent ones. Calcarine fissure is the primary visual area with gyrus above and below it. The auditory area, I would say the cingulate area, I forgot about that. The cingulate area is this right here. We're going to talk about the cingulate gyrus at the end because it has a special function. It's heavily involved in the limbic system, that one particular gyrus of cortex. Area 41 and 42 is the transverse gyrus of Heschel. We know that as the primary auditory area. And then you have 22, which is over here, right there. And then 44 and 45, which is right here. It's right in front of the precentral gyrus, right above the lateral fissure. There's the precentral gyrus. It's right in front. These areas are known respectively as Wernicke's area and Broca's area. Broca's area, as it says here, is your motor speech area. It drives your vocal folds. Now, connections from the cortex. Oh, wow, this is a hot mess. Forget all of this stuff up here. Forget all of this stuff up here. Just look at this. Look at this. because you know this already. What is the input to the cerebral cortex? I'm going to talk to you in just a second about association fibers. That's just one area of the cortex talking to another. Callosal. Callosal is one side of the cortex talking to the other side. This is the corpus callosum right here, this large mass of fibers here. Dr. Morgan mentioned it yesterday. The corpus callosum connects the right brain to the left brain. And then you've got the thalamocortical. This is what you know. Thalamocortical, we know that the thalamus projects to the cortex. We know that. Right? Now, output, we come over here, you know these. Association and callosal, obviously, you've got the association between the two and then across the sides. Then you've got corticospinal, we know that. Corticofugal, corticofugal is another word for anything coming out of the a projection neuron, like the corticobulbar to the brainstem. That's fugal just means a projection axon. And then corticothalamic. We know that the cortex projects to the thalamus. That's it. It's that simple. So let's look at certain neurotransmitters that affect the cortex. Not that they are in the cortex as neurons, but they project to the neocortex in particular. One is dopamine. Dopamine, we know, comes from the caudate, the substantia nigra. There are all sorts of projections from the brainstem up to the cortex. If you have too little dopamine in your system, that's Parkinson's disease. If you have too much, it's schizophrenia. That's speaking very generally, but that's basically it. Drugs to treat schizophrenia impact dopamine. Noradrenergic is responsible for the plasticity of the brain. Right down in here, and I didn't talk about this, but somebody mentioned it, and that is when you're asleep, how do you wake up? How do you wake up? Why do you wake up? What tells your brain to wake up?
Sound?
What?
Sound?
No.
Cortisol?
It's this thing. There's a little nucleus down in your brainstem that's called the locus ceruleus. C-E-R-E-U-L-U-S. You don't need to write down. Locus ceruleus is a noradrenergic containing nucleus that just before you wake up, that thing starts firing. It wakes your brain up. That's how you wake up. It's not related to sound. It's not related to anything. Now, things can activate your locus ceruleus like sound. But, you know, I set my alarm clock for 6 o'clock every morning. It's never gone off because I always get up before then. Why? It's that. It's my brain has told my locus ceruleus, start firing at 5:15 or 5:30. I have a friend that has never used an alarm clock in his life. If he knows he's got to get up at 4 o'clock the next morning to go fishing, he gets up at 4 o'clock. He wakes up at 4 o'clock. If he has to get up, if he can sleep till 7 to be somewhere, he doesn't even own an alarm clock. Kendall.
So what happens with people that have a really hard time waking up or sleep through their alarms?
They're mostly just students. Yeah. It has to do like with peeing in your pants and development of myelin. When you grow up, so yeah, I don't know. I don't know why. But if you don't set an alarm clock and you have no test on Monday and you're going to sleep in on Saturday and you don't wake up until 10 o'clock, why did you wake up at 10 o'clock? It was your locus ceruleus saying, you know, Kendall, you're going to get decubiti if you don't get out of here. So, anyway, that's the noradrenergic. And a lot of the learning in the brain, as it says here. Serotonergic pathways, you know about serotonin and depression, SSRIs. Too little is that too much serotonin in your system produces a thing called serotonin syndrome, where it impacts your autonomic nervous system. It activates your autonomic nervous system. Your blood pressure goes way up. Your heart rate goes way up. You sweat like crazy. It can actually be life-threatening. So you cannot combine a lot of serotonin-promoting drugs. You can't combine two serotonin-promoting drugs because of the possibility of serotonin syndrome. The cholinergic pathways we know all about are related to dementia, Alzheimer's, and memory. Now, this is an illustration of the cortical columns that I mentioned the other day regarding the visual cortex. They put a radio label in the eyeball of one animal, like a monkey or an experimental animal, and that radio active substance is picked up and travels back to the cortex over time. And then they do a study where they put a photographic film on it, develop the film, and then you know where the radioactivity is. And you can see the columns of neurons related to that one eyeball. So this would be your right eye, left eye, right eye, left eye, right eye, left eye. That's how the cortical columns are arranged. Okay? Parts of your nervous system have different functions based on how you're using them. This right here deals with just language. PET scanning, the way you do a PET scan is you take glucose and you radio label it. Then you inject that into a person, and neurons with high levels of activity take up that glucose. And then you put them on a scanner that's essentially a Geiger counter, and it colorizes the areas of highest activity. And if you look at the cortex, depending on how you're using your brain, different areas will light up, even if it's for the same purpose. And this is speech or language. Whether or not you're talking, that's Broca's area right there, the motor speech area. This is generating verbs here. That's having to think about the context of words, or the syntax, right? Listening to words involves the transverse gyrus ephemeral, and viewing words is back here in the occipital cortex. Kind of cool, huh? All with the same thing, all dealing with the same thing. Language is just the different parts of your nervous system taken care of. So, here's the thing regarding the cerebral cortex. Everything you think you hear, wait a minute, why am I not just saying everything you hear? Why am I not saying that? Because you think you hear something, an auditory hallucination. You know what presbycusis is? Tinnitus. I have this loud, high-pitched ringing in this ear right now. I can hear it. It's loud. Can you hear it? Can you not hear it? How can you not hear this loud sound over here? I think I hear it, but it's not really there. That's why I say everything you think you hear. Because what I hear and what you hear, two different things. Everything you think you see. What numbers do you see here?
74, 73.
I don't see a single one of them. I do not see a single number up there. I'm red, green, colorblind. Everything you think you see, I don't see a thing. You're sitting there, oh, there's 22, 26. Those numbers are not there. We're looking at the same thing. It's not everything we see, it's everything we think we see. Okay? Same with taste and smell. Same with everything. Your sense of self, everything you feel you are. That's interesting. I mean, you gotta really look at this, you know? It's not a matter of just neural transmission. It's how you, where you are with it. That's the difference between what constitutes the brain and what constitutes the mind. There's a dichotomy there between the two. And it's stated right here. The mind is what makes you you, or makes us human, separates us from the lower phylogenetic tree. Like this. These are the things that, when you go away from humans, they don't do. Or they at least don't do to the extent that we do. Now, there's a little bit of, what's it called, anthropomorphic thinking here. Because you don't know if a great ape has spirituality. You don't know if a orangutan believes in a God. That's what we think that they don't do. But we don't really know that, do we? So, why don't you go activate your micturition reflex, and then we'll come back and